We've also seen some data for nintedanib, at least last year. TKI, again, a pure VEGF targeted agent, which while the trial wasn't positive overall, at least in the non-squamous cohort, again, in the similar setting, docetaxel plus or minus, suggested in a post-hoc analysis, it was actually statistically significant improvement. Any comments there?
If I recall, it was a relatively modest benefit, and
2 months.
It didn't make me excited about its potential.
And I believe it inhibits more than VEGFR. I believe it hits FGFR as well as PDGFR.
There is a phase 3 study going to be done in the Sun.
They're going to repeat the study strictly in non-squamous.
Population. But I agree with Ross. I think unless we select a group of patients which would respond to this, who would respond to this combination, these kinds of unselected group of patients, it's unlikely that this kind of studies, the unselected group of patients would.
In 12 years, we do not have a viable marker for the benefit of angiogenesis. Any progress there, Ross?
Well, actually, no. I just wanted to jump on something that Alice said, which I thought was a very good point, is that this isn't just a VEGFR inhibitor. We now know that, for example, FGFR, there's a proportion of people who may be sensitive to FGFR inhibitors. They may not have the best biomarker, but certainly we're seeing, lowish percentage response rates to FGFR inhibitors and FGFR amplified squamous. So when you have a drug that, hits one of those targets and you have this unselected population, you don't know if that apparent 2 months benefit is because you're really working on a rather small population within there.
I absolutely agree with Ross, and we do see PDGFR mutations in lung cancer, and this dramatic benefit in small number of patients may be swept away by the lack of response in others, and they never find this. I think the companies ought to do a better job in identifying the group that would respond to and developing trials in the population.
So really identifying these relatively infrequent, more obscure molecular abnormalities and targeting patients for that.
But it's a challenge from a drug development point of view because it's very costly to do these sorts of things. There's a high degree of risk. And the ultimate payoff may be you may find a good drug for a relatively small patient population. And
but say that to the ALK developers, 1%, 2%, 3% of the population. Now we have 10 drugs. So I think it's possible if you do it.
I think you've brought up a major issue, which is certainly a focus in squamous cell, the master lung protocol. You want to describe that effort and where we are with squamous histology?
I think the squamous cell lung cancer, we have learned from the Cancer Genome Atlas work and others that nearly 2/3 of them are potentially actionable targets. And the issue really is we say potentially actionable means we think we can target these genes. But then we don't have those drugs well defined yet. And I think the idea is how do you develop these trials? And that's the whole idea of this master protocol where you will have a control arm, you will have a group of well-defined population, and a targeted agent will be selected and will be tested in that population. And then you have multiple different arms going on. When you have this multi-gene testing and you find that somebody has X mutation, you have a specific arm you can go to, or if you have Y mutation, you can go to the different group. And the whole idea is to build a nice scaffold or infrastructure through which we can study these agents in a continual fashion rather than opening and closing these studies. And I think this is the great model. This is the way I think we can develop these rare genotype-directed trials in a rapid, high-throughput fashion.
And certainly squamous cell is a major unmet need. We don't have any targeted therapy for that.
Without a question. And I think we have badly scratched the surface. After all, we have done only the DNA sequencing. We are beginning to do RNA sequencing. There are tons of fusion genes that I don't think we have identified yet. ALK is an example. ROS is an example. In squamous cell, they have FGFR TAC1 fusion, and I think that's a great example. And I think we'll find more and more of that as we study with the transcriptome sequencing, and I think we need to be looking at that very carefully.
But I think we need to recognize one recent advance in squamous, and that's the positive phase 3 trial with nesitubimab. Nesitubimab is the fully humanized cetuximab. I think most people are familiar with the FLEX data in the the marginal benefit we saw, even though it was a positive phase 3 trial.
It never led to a formal approval.
Never led to a formal approval, and I think most oncologists were not necessarily blown away with the benefit that was seen in the FLEX trial. Necitumumab is a different molecule. This trial was done in, the SQUIRE trial was done in a squamous-only population, but 1,050 patients, in which the design was cis-gemcitabine plus or minus necitumumab. There were no selection criteria based on protein expression, so this was all comers with squamous, and there was an overall survival advantage seen with the addition of this antibody to a very standard option for squamous cell patients. So I think we'll see that data at ASCO 2014 and you know, see if that really represents an advance. I think that this is a meaningful advance only in the sense that our options for the squamous have been really limited. And the magnitude of the benefit, I think, is clinically relevant in this population.
So statistically significant and clinically meaningful, not
Yeah, well,
just marginal.
You can see the abstract and see the presentation and decide for yourself.
The original FLEX trial